Hi, my name is Aishi, and today we'll be talking about bulk transport. And we know that it's pretty easy to move small molecules in and out of the cell because if they're small and nonpolar enough, they can easily diffuse, and if they are, not, then they can be easily pumped across the membrane with transport proteins, either passively if it's going along a gradient of some sort, or actively with use of ATP. But it's much harder to move large molecules such as proteins and polysaccharides in this way. And so what the cell has to do is use vesicles to move these large molecules in and out of the cell. And as you might imagine, this uses up energy because you have to physically move these huge things around the cell and have the vesicles fuse with the plasma membrane or form the vesicles from the plasma membrane itself. So it makes sense that it uses energy. Now you can either have movement out of the cell and movement into the cell. So movement out is called exocytosis, and movement in is called endocytosis. Now remember, exocytosis is movement out of the cell. So the cell uses exocytosis when it wants to release some biological molecules into the surroundings of the cell. For example, if it is one of the cells in the pancreas, it might use exocytosis to secrete insulin. And if it's a neuron, it could use exocytosis to release neurotransmitters and have that signal to other neurons. So here I have drawn a diagram of a cell. And I drew the plasma membrane in two colors, green and blue. So the outer membrane, or the extracellular membrane of that bilayer, is green, and the inner membrane, or the side facing the cytoplasm, is in blue. And now, as the vesicle buds off from the Golgi apparatus, and it contains the molecules to be released into the environment, notice the colors of the two layers. The outer layer is blue and the inner layer is green. And the reason for this is because when the vesicle fuses with the plasma membrane, essentially it all reverses. So the inside of the vesicle, which is green here, actually becomes the outside of the plasma membrane. So green, green, blue, blue, and the outside of the vesicle becomes merged with the inside of the plasma membrane. So the color coding makes sense. So now let's talk about endocytosis. Remember that endocytosis is movement into the cell. Now there are three types that we'll be talking about today: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Now phagocytosis is essentially cellular eating. We have a food particle here, and as you can see, what the cell does is it reaches out with this plasma membrane and wraps this plasma membrane around this food particle or large molecule, whatever, and it engulfs it and forms a vesicle and moves it into the cell. Now these projections are temporary and they're called pseudopods. The word pseudopod in Greek means false foot, and it is a false foot. It's temporary. It doesn't exist in the long term. Now pinocytosis is cellular drinking. What the cell is trying to do is it cares about these molecules that are dissolved within the extracellular fluid, and is trying to get these guys into the cell. And so what it does is it pinches off part of its plasma membrane and forms a vesicle. And the vesicle would include part of the extracellular fluid, and along with it, these molecules that are dissolved within it. Now, receptor-mediated endocytosis is a great way of making bulk transport more specific. So let's say you have a molecule, and there's very little of this molecule that's dissolved within the extracellular fluid. Now, it would be really inefficient to just do pinocytosis a lot of times and continually just taking up extracellular fluid, because you're not going to get very much of this molecule that you want. And so what the cell does is it has receptors located in the plasma membrane, and when the molecule of interest binds to these receptors will gather together and bud off and form a vesicle, as shown here.